Hey, this is Mr. Peterson. This screencast is about some membranes, or just membranes in general. And membranes are barriers that really can isolate things. And so they separate different parts of a cell and different parts of what goes on inside of a cell. Here's a membrane. This is an amoeba. And this is in real time. And that's a membrane. Look at how fluid and flexible it is as this amoeba scooches all over the place. So amoeba are freshwater organisms that have the ability to move. We have cells in our body that can do that. Let's talk about the components, the structure of a membrane. First of all, membranes are all of them, and it doesn't matter which membrane it is. If it's a cellular membrane or a membrane that's inside of or around a structure inside of a cell, they all are composed of phospholipids. So here's a quick picture, and they have two main functions. Number one, they form a boundary, so a barrier to something, and then they control what goes through that boundary, what goes in and out of that membrane. So inside and outside of the cell, we can see that a cell membrane has the control of that. So phospholipids make up all membranes. So phospholipids have two important parts to them. They have a polar head, so that's the orange glob, and then the long non-polar fatty acid tails. So we've looked at this. So here's the flexible fatty acid tails and the non-polar head. It's a phosphate, so they tend to have a phosphorus atom and 4 oxygen surrounding them, and they have a charge to them, so they're polar. They can interact with water. The fatty acids, not so much. But phospholipids make up all membranes with that basic component. Now, if you look at this, here's a phospholipid bilayer, and you can see that it's moving. And so those blue things, at least I think they're blue, those are the fatty acids. And then the yellow things, ooh, do you recognize those from that packet of biomolecules? Yeah, those are cholesterol molecules. Those are steroids. And steroids and cholesterols are important components of membranes. Helps keep them fluid. So phospholipid bilayers, we got other molecules that are embedded into the membrane, and they're going to have important functions as far as regulating what goes in and out. And then we have what's called scientists have adapted to what's called a fluid mosaic model. So think about a mosaic. It's composed of a lot of different pieces, and in this case, it's fluid. So think about the soap films and playing with those and how fluid those were. So here's an example of a phospholipid bilayer inside and outside of the cell. I'm going to kind of scroll through this kind of quickly. So it has that phospholipid layer. And notice that the fatty acid tails move. So they're oily. Think about that. They're not static thing. And we have two important terms here, hydrophilic and hydrophobic. What does that mean, hydrophilic? Hydrophobic. I think you probably know about phobias. Phobias are fears. So the fats, those are hydrophobic. They're afraid of water. The other end, the polar end, likes water. So hydrophilic. Philios means love. So a philia is kind of the opposite of a phobia. Now there are other things embedded in there. So here's some integral membrane proteins, which means they're right in the membrane, and actually they go all the way through. We have some that are kind of stuck on the outside. We also have sugars that will act as kind of like, hey, we're kind of cell markers. We're identifying the cells. Those are oligosaccharides, so you've seen the word monosaccharide or polysaccharide. And then cholesterols, which help keep the membranes fluid. So membranes are really complex, the fluid mosaic model. They're also selectively permeable. I think the word selective is pretty evident, but permeability, that might be new to you. So permeability is like the ability to move through something. So selectively permeable. Now, what do you think are some limits to what can travel through this phospholipid bilayer? What would be some things? What would be some limitations? So think about that for a second. Well, there are a couple of basic ideas. Number one, if things are ionic, if things have charges, they can be attracted to this part, but they're repulsed they're pushed back on the lipid part, so they can't pass. So ions. So remember these guys, sodium and chloride ions, they can't pass through. Little tiny molecules like water, they can kind of sneak through a little bit. Plus there's some other structures, some pores in there. So size, charge, whether things are polar or nonpolar, those are some things that are going to determine whether something can pass through a membrane. So it's selectively permeable. Not everything can pass through a membrane, and that's a good thing because that really helps regulate what goes in and out of cells and in and out of membranes that are around things inside of cells. So selective permeability. So small things can pass through pretty easily. Okay, so just a quick review. Membranes have components, mostly phospholipids, fatty acid tail, polar head, and they're inside the cell. They're surrounding a cell, but they're also around structures inside of cells. They're made of phospholipids. We have the hydrophilic, which are the water-loving ends. That's the phosphate group. The ping pong ball likes water. It's hydrophilic. Then the ping pong ball likes water. The hydrophobic end, the fatty acid tails, they repel water. They don't like water. So water, if water is going to go through a membrane, it's going to use some special structures called aquaporins. We'll get to that in a different screencast. They're very fluid. They move. We saw that with the amoeba. And they're selectively permeable. So some stuff about membranes. I hope that helps.